Welcome to this comprehensive journey through the entire biology of the human body. As you rest and listen, allow yourself to absorb the fascinating intricacies of how your body functions at every level. From the smallest cellular components to the largest organ systems working in perfect harmony. The human body is an extraordinary biological machine, comprising approximately 37 trillion cells, each performing specific functions that contribute to your survival and well-being. Let us begin this exploration at the most fundamental level, examining the building blocks that make you who you are. Every cell in your body contains a nucleus, which houses your genetic material in the form of DNA, deoxyribonucleic acid, a molecule that carries the complete instructions for building and maintaining your entire body. This DNA is organized into 46 chromosomes, 23 inherited from each parent, containing approximately 20,000 genes that code for proteins essential to life. The cell membrane, a phospholipid bilayer, surrounds each cell, controlling what enters and exits while maintaining the delicate internal environment. Within the cytoplasm, numerous organelles perform specialized tasks. Mitochondria, often called the powerhouses of the cell, generate ATP through cellular respiration, converting glucose and oxygen into usable energy. The endoplasmic reticulum, both rough and smooth varieties, manufactures proteins and lipids, while the Golgi apparatus packages and distributes these molecules throughout the cell. Ribosomes, either free-floating or attached to the rough endoplasmic reticulum, translate messenger RNA into proteins through a process involving transfer RNA and amino acids. Lysosomes contain digestive enzymes that break down cellular waste and foreign materials, maintaining cellular health. The cytoskeleton, composed of microfilaments, intermediate filaments, and microtubules, provides structural support and enables cellular movement and division. Now let us ascend from the cellular level to tissues, which are groups of similar cells working together to perform specific functions. The human body contains 4 primary tissue types. Epithelial tissue covers body surfaces, lines hollow organs and cavities, and forms glands. This tissue provides protection, absorption, filtration, and secretion. Simple epithelium consists of a single layer of cells and is found in areas where absorption or filtration occurs, such as the lining of blood vessels and air sacs in the lungs. Stratified epithelium has multiple layers and provides protection in areas subject to abrasion, like the skin and esophagus. Connective tissue supports, binds, and protects other tissues and organs. It includes bone, cartilage, adipose tissue, blood, and loose and dense connective tissues. The extracellular matrix in connective tissue contains various fibers including collagen for strength, elastic fibers for flexibility, and reticular fibers for support. Muscle tissue enables movement through contraction. Skeletal muscle attaches to bones and produces voluntary movements. These muscles are striated, showing alternating light and dark bands under microscopy, resulting from the organized arrangement of actin and myosin filaments. Cardiac muscle, found only in the heart, is also striated but operates involuntarily, controlled by the heart's intrinsic pacemaker cells. Smooth muscle lacks striations and is found in the walls of hollow organs like the stomach, intestines, and blood vessels, controlling involuntary movements such as peristalsis and vasoconstriction. Nervous tissue consists of neurons and supporting glial cells. Neurons transmit electrical signals throughout the body, enabling communication between different body parts and the brain. Each neuron has a cell body containing the nucleus, dendrites that receive signals, and an axon that transmits signals to other cells. Glial cells provide support, nutrition, and protection to neurons. Moving to the organ system level, let us explore the integumentary system, your body's largest organ system. The skin covers approximately 2 square meters in adults and serves multiple critical functions. The epidermis, the outermost layer, consists primarily of keratinocytes that produce keratin, a tough protective protein. The deepest layer of the epidermis, the stratum basale, contains stem cells that continuously divide to replace dead skin cells shed from the surface. Melanocytes in this layer produce melanin, the pigment responsible for skin color and protection against ultraviolet radiation. The dermis beneath contains blood vessels, nerve endings, hair follicles, and glands. Sebaceous glands secrete sebum, an oily substance that lubricates skin and hair, while sweat glands help regulate body temperature through evaporative cooling. The hypodermis, or subcutaneous layer, consists mainly of adipose tissue that insulates the body and anchors the skin to underlying structures. Hair grows from follicles embedded in the dermis, with each strand composed of dead keratinized cells. Nails, protective coverings on fingers and toes, also consist of keratin and grow from the nail matrix at their base. The skeletal system provides structure, protection, and support, while enabling movement and producing blood cells. An adult human skeleton contains 206 bones, though infants have approximately 300, many of which fuse during development. Bones are living tissues constantly undergoing remodeling through the coordinated action of osteoblasts, which build new bone, and osteoclasts, which break down old bone. This process allows bones to adapt to mechanical stress and repair damage, while also regulating calcium homeostasis in the blood. Bone tissue exists in 2 forms. Compact bone, dense and strong, forms the outer layer of all bones and the shaft of long bones. Spongy bone, with a honeycomb-like structure, is found at the ends of long bones and in the interior of other bones, providing strength with reduced weight. Red bone marrow within spongy bone produces blood cells through hematopoiesis, generating millions of new red blood cells, white blood cells, and platelets every second. Yellow bone marrow consists primarily of adipose tissue and serves as an energy reserve. The skeleton divides into the axial skeleton, comprising the skull, vertebral column and rib cage, and the appendicular skeleton, consisting of the limbs and the girdles that attach them to the axial skeleton. The skull protects the brain and consists of 22 bones, including 8 cranial bones and 14 facial bones. The vertebral column contains 33 vertebrae, though some fuse in adults, leaving 26 separate bones. Between vertebrae, intervertebral discs composed of fibrocartilage absorb shock and allow flexibility. The rib cage protects vital organs in the thoracic cavity, with 12 pairs of ribs attaching to the thoracic vertebrae. Joints where 2 or more bones meet are classified by structure and function. Fibrous joints, like those between skull bones, allow little or no movement. Cartilaginous joints, such as intervertebral discs, permit limited movement. Synovial joints, the most common and movable type, feature a joint cavity filled with synovial fluid that lubricates the joint and nourishes the articular cartilage covering bone ends. Examples include the knee, elbow, and shoulder joints. Ligaments, tough bands of dense connective tissue, connect bones to bones, providing stability while allowing appropriate movement. The muscular system works intimately with the skeletal system to produce movement, maintain posture, and generate heat. The human body contains over 600 muscles, accounting for approximately 40% of body weight in males and slightly less in females. Skeletal muscles attach to bones via tendons, strong cords of dense connective tissue. Muscle contraction occurs through the sliding filament mechanism, where myosin heads attach to actin filaments and pull them toward the center of the sarcomere, the basic contractile unit. This process requires ATP and is triggered by calcium ions released from the sarcoplasmic reticulum in response to nervous stimulation. The neuromuscular junction, where a motor neuron meets a muscle fiber, transmits signals via the neurotransmitter acetylcholine. Each motor neuron and all the muscle fibers it innervates constitute a motor unit. Muscles work in antagonistic pairs, with one muscle contracting while its partner relaxes. For instance, the biceps brachii flexes the elbow while the triceps brachii extends it. Muscles are named based on various characteristics including their location, shape, size, direction of fibers, number of origins, points of attachment, and action. The nervous system, an intricate network of cells and tissues, coordinates body activities and enables you to perceive and respond to your environment. It divides functionally into the central nervous system, comprising the brain and spinal cord, and the peripheral nervous system, consisting of all nerves extending from the central nervous system to the rest of the body. The brain, weighing approximately 1.4 kilograms, contains roughly 86 billion neurons. The cerebrum, the largest part, divides into left and right hemispheres connected by the corpus callosum, a bundle of nerve fibers enabling communication between hemispheres. The cerebral cortex, the outer layer of gray matter, contains neuron cell bodies and is responsible for higher cognitive functions including consciousness, thought, memory, and voluntary movement. The cortex organizes into lobes with specialized functions. The frontal lobe controls executive functions, decision-making, planning, and voluntary movement through the primary motor cortex. The parietal lobe processes sensory information related to touch, temperature, pain, and proprioception. The temporal lobe handles auditory processing, language comprehension, and memory formation, housing the hippocampus, crucial for converting short-term memories to long-term storage. The occipital lobe processes visual information from the eyes. Beneath the cortex, white matter consists of myelinated axons transmitting signals between different brain regions. Deep within the cerebrum lie the basal ganglia, involved in movement control and procedural learning, and the limbic system, which processes emotions and memory. The thalamus acts as a relay station for sensory information, while the hypothalamus regulates homeostasis, controlling body temperature, hunger, thirst, sleep-wake cycles, and the endocrine system through its connection to the pituitary gland. The cerebellum, attached to the brainstem, coordinates voluntary movements, maintains balance and posture, and helps with motor learning. The brainstem, consisting of the midbrain, pons, and medulla oblongata, controls vital autonomic functions including heart rate, breathing, and blood pressure, while also serving as a pathway for signals between the brain and spinal cord. The spinal cord extends from the medulla oblongata through the vertebral canal to the lumbar region. Protected by cerebrospinal fluid and the meninges, 3 protective membranes, it serves as a conduit for signals between the brain and peripheral nerves and coordinates reflex actions. 31 pairs of spinal nerves emerge from the cord, each containing both sensory and motor neurons. The peripheral nervous system further divides into the somatic nervous system, controlling voluntary movements and conveying sensory information to the central nervous system, and the autonomic nervous system, regulating involuntary functions. The autonomic system subdivides into sympathetic and parasympathetic divisions that generally have opposing effects. The sympathetic division prepares the body for stressful or emergency situations, the fight-or-flight response, increasing heart rate, dilating airways, and redirecting blood to muscles. The parasympathetic division promotes rest and digest activities, slowing heart rate, stimulating digestion, and conserving energy. Neurons communicate through synapses, tiny gaps between cells where neurotransmitters carry signals from one neuron to another. When an action potential reaches the axon terminal, voltage-gated calcium channels open, allowing calcium influx that triggers synaptic vesicles to release neurotransmitters into the synaptic cleft. These molecules bind to receptors on the postsynaptic neuron, either exciting or inhibiting it. Major neurotransmitters include acetylcholine, involved in muscle contraction and memory, dopamine, associated with reward and movement, serotonin, regulating mood and sleep, norepinephrine. Involved in arousal and alertness, GABA, the primary inhibitory neurotransmitter, and glutamate, the primary excitatory neurotransmitter. The endocrine system works alongside the nervous system to regulate body functions through chemical messengers called hormones. Unlike the rapid electrical signals of the nervous system, hormones travel through the bloodstream, producing slower but longer-lasting effects. The pituitary gland, often called the master gland, sits beneath the hypothalamus and secretes hormones controlling other endocrine glands. The anterior pituitary produces growth hormone, stimulating growth and cell reproduction, thyroid-stimulating hormone, regulating thyroid function, adrenocorticotropic hormone, controlling cortisol release from adrenal glands, follicle-stimulating hormone and luteinizing hormone, regulating reproductive functions, and prolactin, stimulating milk production. The posterior pituitary stores and releases oxytocin, which stimulates uterine contractions during childbirth and milk ejection during breastfeeding, and antidiuretic hormone, which regulates water balance by promoting water reabsorption in the kidneys. The thyroid gland in the neck produces thyroid hormones that regulate metabolism, growth, and development, and calcitonin, which helps regulate calcium levels. The parathyroid glands, 4 small glands on the thyroid's posterior surface, secrete parathyroid hormone, which increases blood calcium levels by stimulating bone resorption, enhancing calcium absorption in the intestines, and promoting calcium reabsorption in the kidneys. The adrenal glands, sitting atop the kidneys, consist of an outer cortex and inner medulla. The cortex produces corticosteroids including cortisol, which regulates metabolism and stress response, aldosterone, which controls sodium and potassium balance, and small amounts of sex hormones. The medulla secretes epinephrine and norepinephrine in response to stress, mediating the fight-or-flight response. The pancreas functions as both an endocrine and exocrine gland. Its endocrine portion, the islets of Langerhans, contains beta cells producing insulin, which lowers blood glucose by promoting cellular uptake and storage, and alpha cells producing glucagon, which raises blood glucose by stimulating glycogen breakdown and glucose synthesis in the liver. The gonads, ovaries in females and testes in males, produce sex hormones in addition to gametes. Ovaries secrete estrogen and progesterone, regulating the menstrual cycle, pregnancy, and secondary sexual characteristics. Testes produce testosterone, controlling sperm production and male secondary sexual characteristics. The pineal gland secretes melatonin, regulating sleep-wake cycles in response to light-dark cycles. The thymus, active mainly in childhood, produces hormones supporting T-cell maturation and immune function. The cardiovascular system, consisting of the heart, blood vessels, and blood, transports oxygen, nutrients, hormones, and waste products throughout the body. The heart, a muscular organ roughly the size of a closed fist, beats approximately 100,000 times daily, pumping about 5 liters of blood per minute at rest, increasing dramatically during exercise. The heart has 4 chambers, 2 upper atria that receive blood and 2 lower ventricles that pump blood. The right atrium receives deoxygenated blood from the body through the superior and inferior vena cava. This blood flows into the right ventricle, which pumps it through the pulmonary arteries to the lungs for oxygenation. Oxygenated blood returns to the left atrium via pulmonary veins, flows into the left ventricle, and is pumped through the aorta to the rest of the body. Heart valves ensure unidirectional blood flow. The tricuspid valve lies between the right atrium and ventricle, the pulmonary valve between the right ventricle and pulmonary artery, the mitral or bicuspid valve between the left atrium and ventricle, and the aortic valve between the left ventricle and aorta. The cardiac cycle consists of systole, when ventricles contract and eject blood, and diastole, when ventricles relax and fill with blood. The heart's rhythmic contractions originate from the sinoatrial node, the natural pacemaker in the right atrium, which generates electrical impulses spreading through the atria, causing them to contract. The impulse reaches the atrioventricular node, pauses briefly allowing the atria to finish contracting, then travels through the bundle of His and Purkinje fibers, triggering ventricular contraction. Cardiac muscle cells are connected by intercalated discs containing gap junctions that allow electrical signals to spread rapidly throughout the heart muscle. Blood vessels form an extensive network extending approximately 100,000 kilometers if laid end to end. Arteries carry blood away from the heart under high pressure, with thick, elastic walls allowing them to stretch with each heartbeat and maintain blood flow between contractions. The largest artery, the aorta, branches into smaller arteries, then arterioles, which regulate blood flow to capillary beds through vasoconstriction and vasodilation. Capillaries, the smallest blood vessels with walls only 1 cell thick, facilitate exchange of oxygen, nutrients, and waste products between blood and tissues. Blood flows from capillaries into venules, then into larger veins that return blood to the heart. Veins have thinner walls than arteries and contain valves preventing backflow, especially in the limbs where blood must flow against gravity. Skeletal muscle contractions and breathing movements help propel blood through veins. Blood, a connective tissue consisting of cells suspended in plasma, comprises about 8% of body weight. Plasma, the liquid component making up 55% of blood volume, is 90% water and contains proteins, nutrients, hormones, gases, and waste products. The formed elements, or blood cells, include red blood cells, white blood cells, and platelets. Red blood cells, or erythrocytes, are the most numerous, with approximately 5 million per microliter of blood. These biconcave disc-shaped cells lack nuclei, maximizing space for hemoglobin, the iron-containing protein that binds and transports oxygen. Each hemoglobin molecule can carry 4 oxygen molecules. Red blood cells are produced in red bone marrow and circulate for about 120 days before being removed by the spleen and liver. The kidneys produce erythropoietin, a hormone stimulating red blood cell production in response to low oxygen levels. White blood cells, or leukocytes, defend against infection and disease. There are several types, each with specific functions. Neutrophils, the most abundant, are phagocytes that engulf and destroy bacteria and cellular debris. Eosinophils combat parasitic infections and play a role in allergic reactions. Basophils release histamine and heparin, involved in inflammatory responses. Lymphocytes include B cells, which produce antibodies, T cells, which directly attack infected cells and coordinate immune responses, and natural killer cells, which destroy virus-infected and cancerous cells. Monocytes circulate in blood and differentiate into macrophages when they enter tissues, where they phagocytose pathogens and cellular debris. Platelets or thrombocytes are cell fragments essential for blood clotting. When a blood vessel is damaged, platelets adhere to the exposed collagen, become activated, and release chemicals attracting more platelets, forming a platelet plug. They also initiate the coagulation cascade, a series of reactions involving clotting factors that culminate in the conversion of fibrinogen to fibrin, forming a mesh that stabilizes the platelet plug into a clot. The respiratory system enables gas exchange, bringing oxygen into the body and expelling carbon dioxide, a waste product of cellular metabolism. Air enters through the nose or mouth, where it is filtered, warmed, and humidified. The nasal cavity contains mucus-secreting goblet cells and cilia that trap and remove particles. Air passes through the pharynx, a passageway shared with the digestive system, then enters the larynx, which contains the vocal cords. The epiglottis, a flap of cartilage, covers the larynx during swallowing, preventing food from entering the airways. The trachea, or windpipe, is reinforced with C-shaped cartilage rings maintaining its patency. It divides into left and right primary bronchi, each entering a lung. The bronchi branch repeatedly into smaller bronchioles, forming a tree-like structure. The smallest bronchioles terminate in alveoli, tiny air sacs where gas exchange occurs. The lungs contain approximately 300 million alveoli, providing a surface area of about 70 square meters for gas exchange. Alveolar walls are extremely thin, consisting of a single layer of epithelial cells surrounded by a network of capillaries. Oxygen diffuses from alveoli into blood, while carbon dioxide diffuses from blood into alveoli. Type 2 alveolar cells produce surfactant, a substance reducing surface tension and preventing alveolar collapse. The pleural membranes, 2 layers surrounding each lung with a fluid-filled space between them, reduce friction during breathing and help maintain lung inflation. Breathing, or ventilation, involves inhalation and exhalation. During inhalation, the diaphragm, a dome-shaped muscle separating the thoracic and abdominal cavities, contracts and flattens, while external intercostal muscles contract, elevating the ribs. These actions increase thoracic cavity volume, decreasing pressure and drawing air into the lungs. During exhalation, the diaphragm and intercostal muscles relax, thoracic volume decreases, pressure increases, and air is expelled. At rest, exhalation is passive, but during forced breathing, internal intercostal muscles and abdominal muscles contract to further decrease thoracic volume. The respiratory center in the medulla oblongata controls breathing rhythm by monitoring blood carbon dioxide levels. Increased carbon dioxide stimulates deeper, faster breathing to expel excess carbon dioxide and bring in more oxygen. The digestive system breaks down food into nutrients that can be absorbed and used by the body, while eliminating indigestible waste. Digestion begins in the mouth, where mechanical breakdown occurs through chewing, and chemical digestion starts with salivary amylase, an enzyme in saliva that begins breaking down carbohydrates. The tongue manipulates food, mixing it with saliva to form a bolus that can be swallowed. During swallowing, the tongue pushes the bolus into the pharynx, the epiglottis covers the larynx, and peristalsis, wave-like muscle contractions, propels the bolus through the esophagus to the stomach. The lower esophageal sphincter relaxes, allowing the bolus to enter the stomach, then contracts to prevent backflow of stomach contents. The stomach, a muscular sac, stores food and begins protein digestion. Its lining contains gastric glands secreting gastric juice, a mixture of hydrochloric acid and enzymes including pepsin, which breaks down proteins. The acid creates a highly acidic environment, activating pepsin and killing most ingested bacteria. Mucus secreted by the stomach lining protects it from acid damage. The stomach's muscular walls churn food, mixing it with gastric juice to form chyme, a semi-liquid mixture. The pyloric sphincter controls the release of chyme into the small intestine. The small intestine, approximately 6 meters long, is where most chemical digestion and nutrient absorption occur. It consists of 3 sections: the duodenum, jejunum, and ileum. In the duodenum, chyme mixes with bile from the liver and pancreatic juice from the pancreas. Bile, produced by hepatocytes in the liver and stored in the gallbladder, emulsifies fats, breaking them into smaller droplets that enzymes can more easily digest. Pancreatic juice contains bicarbonate, which neutralizes stomach acid, and enzymes that digest carbohydrates, proteins, and fats. The small intestine lining features circular folds, villi, and microvilli, collectively increasing surface area for absorption by about 600 times. Each villus contains a network of capillaries that absorb amino acids and monosaccharides, and a lacteal, a lymphatic vessel that absorbs fatty acids and glycerol. Absorbed nutrients enter the bloodstream and travel to the liver via the hepatic portal vein for processing. The large intestine, approximately 1.5 meters long, consists of the cecum, colon, rectum, and anal canal. Its primary functions are absorbing water and electrolytes, and forming and storing feces. Bacteria in the large intestine, collectively called the gut microbiota, synthesize certain vitamins including vitamin K and some B vitamins, and ferment indigestible carbohydrates. The rectum stores feces until defecation, when the anal sphincters relax and abdominal muscles contract, expelling waste. The liver, the body's largest internal organ, performs over 500 functions. Besides producing bile, it processes nutrients absorbed from the small intestine, storing glucose as glycogen and releasing it when blood glucose levels drop. Converting amino acids and other substances into glucose through gluconeogenesis, synthesizing plasma proteins including albumin and clotting factors, detoxifying drugs and harmful substances, storing vitamins and minerals, and producing cholesterol and lipoproteins for lipid transport. The pancreas, besides its endocrine functions, produces digestive enzymes including pancreatic amylase for carbohydrates, trypsin and chymotrypsin for proteins, and pancreatic lipase for fats. The urinary system maintains homeostasis by regulating blood volume and composition, eliminating metabolic wastes, and controlling electrolyte balance and acid-base balance. It consists of 2 kidneys, 2 ureters, the urinary bladder, and the urethra. The kidneys, bean-shaped organs located in the posterior abdominal wall, filter blood and produce urine. Each kidney contains approximately 1 million nephrons, the functional units where urine formation occurs. Each nephron consists of a renal corpuscle, where filtration occurs, and a renal tubule, where filtrate is modified. The renal corpuscle comprises a glomerulus, a ball of capillaries and Bowman's capsule, which surrounds the glomerulus. Blood enters the glomerulus under high pressure, forcing water and small solutes through the filtration membrane into Bowman's capsule, forming filtrate. Large proteins and blood cells remain in the blood. The filtrate flows through the proximal convoluted tubule, where most water, glucose, amino acids, and ions are reabsorbed into the blood. The loop of Henle creates a concentration gradient in the medulla, enabling the kidney to produce concentrated urine when necessary. The distal convoluted tubule and collecting duct make final adjustments to filtrate composition under hormonal control. Antidiuretic hormone increases water reabsorption, concentrating urine, while aldosterone increases sodium reabsorption and potassium secretion, helping regulate blood pressure and electrolyte balance. The final product, urine, consists of water, urea from protein metabolism, uric acid from nucleic acid metabolism, creatinine from muscle metabolism, and various ions. Urine flows from the collecting ducts through the renal pelvis into the ureters, which use peristalsis to transport it to the urinary bladder, a hollow muscular organ that stores urine. When the bladder fills, stretch receptors signal the brain, creating the urge to urinate. During urination, the detrusor muscle in the bladder wall contracts while the internal and external urethral sphincters relax, allowing urine to flow through the urethra and exit the body. The reproductive system enables the creation of offspring and produces sex hormones influencing many aspects of physiology and behavior. The male reproductive system produces, maintains, and transports sperm and semen, introduces them into the female reproductive tract, and produces male sex hormones. The testes, located in the scrotum outside the body cavity, where temperature is slightly cooler than core body temperature, contains seminiferous tubules where spermatogenesis occurs. Spermatogenesis begins at puberty and continues throughout life, producing millions of sperm daily. Sperm mature and gain motility as they travel through the epididymis, a coiled tube attached to each testis. During ejaculation, sperm travel through the vas deferens, mix with fluids from the seminal vesicles and prostate gland to form semen, and are expelled through the urethra. The seminal vesicles produce a fructose-rich fluid, providing energy for sperm, while the prostate gland secretes a slightly alkaline fluid that helps neutralize acidity in the female reproductive tract. The bulbourethral glands produce a mucus-like fluid that lubricates the urethra. Testosterone, produced by interstitial cells in the testes, promotes sperm production, maintains reproductive organs, and influences secondary sexual characteristics including muscle development, bone density, body hair, and vocal cord thickening. The female reproductive system produces ova, provides an environment for fertilization and fetal development, and produces female sex hormones. The ovaries, located in the pelvic cavity, contain follicles at various stages of development. At birth, ovaries contain approximately 1 to 2 million immature follicles, though only about 400 will mature and release an ovum during a woman's reproductive years. Each month during the menstrual cycle, follicle-stimulating hormone stimulates several follicles to develop, though usually only one becomes dominant. The developing follicle secretes estrogen, which thickens the endometrium, the uterine lining. Around day 14 of a 28-day cycle, a surge in luteinizing hormone triggers ovulation, the release of the ovum from the ovary. The ovum enters the fallopian tube, where fertilization may occur if sperm are present. The ruptured follicle transforms into the corpus luteum, which secretes progesterone and estrogen to maintain the endometrium. If fertilization does not occur, the corpus luteum degenerates, hormone levels drop, and the endometrium is shed during menstruation. If fertilization occurs, the resulting zygote undergoes cell division as it travels down the fallopian tube. About 6 days after fertilization, the blastocyst implants in the endometrium. The outer cells form the placenta, which exchanges nutrients, gases, and wastes between maternal and fetal blood, while also producing human chorionic gonadotropin, which maintains the corpus luteum and prevents menstruation. The inner cell mass develops into the embryo. During pregnancy, the uterus, a pear-shaped muscular organ, expands dramatically to accommodate the growing fetus. The cervix, the lower portion of the uterus, remains tightly closed during pregnancy, protected by a mucus plug. During labor, hormonal changes cause the cervix to dilate and the uterine muscles to contract, eventually expelling the baby through the vagina. The mammary glands, modified sweat glands in the breasts, produce milk after childbirth under the influence of prolactin and other hormones. The immune system defends the body against pathogens, including bacteria, viruses, fungi, and parasites, as well as abnormal cells that could become cancerous. It employs multiple defense mechanisms organized into innate and adaptive immunity. Innate immunity provides immediate, nonspecific defense. Physical barriers form the first line of defense. The skin's keratinized outer layer is impermeable to most microorganisms, while sweat and sebum have antimicrobial properties. Mucous membranes line body cavities open to the exterior, trapping pathogens in mucus that is then expelled. Stomach acid destroys many ingested microorganisms. If pathogens breach physical barriers, cellular and chemical defenses activate. Phagocytes, including neutrophils and macrophages, engulf and destroy pathogens. Natural killer cells destroy virus-infected and cancerous cells by releasing chemicals that induce apoptosis. The complement system, a group of proteins in blood plasma, enhances phagocytosis, promotes inflammation, and directly destroys pathogens by forming membrane attack complexes that puncture cell membranes. Inflammation, characterized by redness, heat, swelling, and pain, is a protective response to tissue injury or infection. Chemical signals from damaged cells and immune cells cause vasodilation, increasing blood flow to the area, and increase capillary permeability, allowing fluid and immune cells to enter tissues. Fever and elevation in body temperature inhibits pathogen growth and accelerates tissue repair. Adaptive immunity provides specific defense against particular pathogens and creates immunological memory, enabling faster, stronger responses upon re-exposure. This system relies on lymphocytes that recognize specific antigens, molecules on pathogen surfaces. B cells mature in bone marrow and are responsible for humoral immunity. When a B cell encounters its specific antigen, it activates, proliferates, and differentiates into plasma cells that secrete antibodies. Antibodies, Y-shaped proteins, bind to antigens, marking them for destruction, neutralizing toxins, preventing pathogen attachment to cells, and activating complement. Some activated B cells become memory cells that persist for years, enabling rapid response if the same pathogen is encountered again. T cells mature in the thymus and mediate cell-mediated immunity. Helper T cells coordinate immune responses by releasing cytokines that activate other immune cells. Cytotoxic T cells directly attack and destroy infected cells and cancer cells by releasing perforins and granzymes that induce apoptosis. Regulatory T cells suppress immune responses, preventing excessive inflammation and autoimmune reactions. Like B cells, some activated T cells become memory cells. The lymphatic system works closely with the immune system, consisting of lymphatic vessels, lymph nodes, and lymphoid organs. Lymphatic vessels collect excess interstitial fluid, now called lymph, and return it to the bloodstream, maintaining fluid balance. Lymph nodes, small bean-shaped structures clustered along lymphatic vessels, filter lymph, trapping pathogens and cancer cells. They contain many lymphocytes and macrophages that respond to trapped pathogens, often swelling during infection. The spleen filters blood, removing old red blood cells and pathogens, and serves as a reservoir for lymphocytes. The thymus, located above the heart, is where T cells mature. Mucosa-associated lymphoid tissue, including the tonsils and Peyer's patches in the small intestine, protects against pathogens entering through mucous membranes. The sensory systems enable you to perceive and interpret information about your environment and internal state. Vision begins when light enters the eye through the cornea, a transparent covering that refracts light. The light passes through the pupil, an opening in the center of the iris, the colored part of the eye that controls pupil size. In bright light, the pupil constricts, reducing light entry. In dim light, it dilates, allowing more light in. The lens, a flexible transparent structure behind the iris, further refracts light, focusing it on the retina at the back of the eye. The ciliary muscles change lens shape, making it more curved for near vision and flatter for distant vision. The retina contains photoreceptors, rods and cones, that convert light into electrical signals. Rods, more numerous and sensitive to low light, enable night vision and peripheral vision, but do not detect color. Cones, concentrated in the fovea, the central area of the retina, require brighter light and enable color vision and detailed central vision. There are 3 types of cones, each sensitive to different wavelengths corresponding to red, green, or blue light. When light strikes photoreceptors, it causes a chemical change in photopigments, triggering a cascade of events that generate electrical signals. These signals are processed by other retinal neurons and transmitted through the optic nerve to the brain. The optic nerves from both eyes meet at the optic chiasm, where fibers from the nasal half of each retina cross to the opposite side, allowing each hemisphere of the brain to receive information from both eyes. Signals travel to the thalamus and then to the visual cortex in the occipital lobe, where they are interpreted as images. Hearing begins when sound waves enter the external acoustic meatus, the ear canal, and strike the tympanic membrane or eardrum, causing it to vibrate. These vibrations transfer to 3 small bones in the middle ear, the malleus, incus, and stapes, collectively called the ossicles. The ossicles amplify the vibrations and transmit them to the oval window, a membrane-covered opening to the inner ear. The inner ear contains the cochlea, a spiral-shaped structure filled with fluid. Vibrations of the oval window create pressure waves in the cochlear fluid, causing the basilar membrane inside the cochlea to vibrate. The organ of Corti, resting on the basilar membrane, contains hair cells with stereocilia that bend in response to membrane movement. This bending opens ion channels, generating electrical signals transmitted via the cochlear nerve to the brainstem, and then to the auditory cortex in the temporal lobe. Different frequencies of sound cause maximum vibration at different locations along the basilar membrane, enabling the brain to distinguish pitch. The inner ear also contains the vestibular apparatus, which detects head position and movement, contributing to balance. The semicircular canals, 3 fluid-filled tubes oriented in different planes, detect rotational movements. When the head rotates, fluid in the canals lags behind, bending hair cells and generating signals about the direction and speed of rotation. The utricle and saccule contain hair cells embedded in a gel-like substance, with calcium carbonate crystals called otoliths. These structures detect linear acceleration and head position relative to gravity. Taste, or gustation, detects chemicals dissolved in saliva. Taste buds, located primarily on the tongue but also on the palate and throat, contain taste receptor cells that respond to 5 basic tastes: sweet, sour, salty, bitter, and umami. A savory taste associated with glutamate. When molecules bind to receptors on taste cells, they generate electrical signals transmitted via cranial nerves to the gustatory cortex. Smell, or olfaction, detects airborne chemicals. Olfactory receptor neurons in the roof of the nasal cavity have receptors that bind to specific odorant molecules. Humans have approximately 400 different olfactory receptor types, enabling us to distinguish thousands of different odors. When odorants bind to receptors, they generate signals transmitted through the olfactory bulb to the olfactory cortex and limbic system, which is why smells can evoke strong memories and emotions. Touch, or the somatosensory system, detects mechanical pressure, temperature, and pain. Various types of receptors in the skin and deeper tissues respond to different stimuli. Mechanoreceptors detect touch and pressure, with some responding to light touch and others to deep pressure or vibration. Thermoreceptors detect temperature changes, with separate receptors for warmth and cold. nociceptors detect painful stimuli including extreme temperature, pressure, and chemical irritants. Proprioceptors in muscles, tendons, and joints detect body position and movement, enabling you to know where your limbs are without looking. Signals from somatosensory receptors travel through peripheral nerves to the spinal cord and ascend to the thalamus, then to the somatosensory cortex in the parietal lobe, which is organized somatotopically, with different regions processing information from different body parts. Your body maintains homeostasis, a stable internal environment despite external changes. Multiple organ systems work together through feedback mechanisms to regulate temperature, pH, blood pressure, blood glucose, and other variables. For example, when body temperature rises, thermoreceptors send signals to the hypothalamus, which initiates cooling mechanisms including vasodilation of skin blood vessels, increasing heat loss through radiation, and activation of sweat glands for evaporative cooling. When body temperature drops, the hypothalamus triggers vasoconstriction to conserve heat and shivering, which generates heat through muscle contraction. Blood glucose regulation involves multiple hormones. After a meal, rising blood glucose stimulates insulin secretion, promoting glucose uptake by cells and storage as glycogen in liver and muscles. Between meals, when blood glucose drops, glucagon secretion stimulates glycogen breakdown and glucose release from the liver. Epinephrine and cortisol also raise blood glucose during stress or fasting. Blood pressure regulation involves baroreceptors in major arteries that detect pressure changes and send signals to the cardiovascular center in the medulla oblongata. If blood pressure rises, the center decreases sympathetic stimulation and increases parasympathetic stimulation, slowing the heart and dilating blood vessels. The kidneys also regulate blood pressure by adjusting blood volume through water retention or excretion, and by secreting renin, which activates the renin-angiotensin-aldosterone system. Increasing blood pressure. Acid-base balance, maintaining blood pH near 7.4, is crucial for protein function and enzymatic activity. The respiratory system regulates pH by controlling carbon dioxide levels, since carbon dioxide combines with water to form carbonic acid. Increased respiration eliminates more carbon dioxide, raising pH, while decreased respiration retains carbon dioxide, lowering pH. The kidneys regulate pH by excreting or retaining hydrogen ions and bicarbonate. Buffer systems in blood, including the bicarbonate buffer system, immediately bind excess hydrogen ions or hydroxide ions, minimizing pH changes. As you rest and absorb this knowledge, appreciate the remarkable complexity and coordination within your body. Trillions of cells work continuously, each performing specialized functions that contribute to your survival and well-being. Organ systems communicate and cooperate, maintaining the delicate balance necessary for life. From the intricate neural networks enabling thought and perception, to the rhythmic contractions of your heart pumping life-sustaining blood. From the exchange of gases in your lungs to the processing of nutrients in your digestive system, every process reflects millions of years of evolution, refined and optimized for human life. Your body constantly adapts to internal and external changes, healing injuries, fighting infections, and maintaining homeostasis. Understanding your body's biology can deepen your appreciation for its capabilities and inspire you to care for it through proper nutrition, exercise, rest, and stress management. Let us continue this journey deeper into the fascinating mechanisms that sustain your existence. The process of cellular respiration, occurring within your mitochondria, is fundamental to life. This metabolic pathway converts glucose and oxygen into carbon dioxide, water, and adenosine triphosphate, the universal energy currency of cells. Cellular respiration consists of three main stages. Glycolysis occurs in the cytoplasm, breaking down one glucose molecule into two pyruvate molecules, generating a net gain of two ATP molecules and two NADH molecules. This ancient metabolic pathway does not require oxygen and likely evolved before oxygen was abundant in Earth's atmosphere. The pyruvate then enters the mitochondria, where it is converted to acetyl coenzyme A, releasing carbon dioxide and generating more NADH. The acetyl coenzyme A enters the citric acid cycle, also called the Krebs cycle, a series of chemical reactions that extract high-energy electrons from the acetyl group, producing carbon dioxide as a waste product and generating ATP, NADH, and FADH2. The electron transport chain, embedded in the inner mitochondrial membrane, is where most ATP is produced. NADH and FADH2 donate electrons to the chain, and as electrons move through protein complexes, energy is released and used to pump hydrogen ions across the membrane, creating an electrochemical gradient. ATP synthase, a remarkable molecular machine, harnesses this gradient as hydrogen ions flow back across the membrane, using the energy to phosphorylate ADP into ATP. Oxygen serves as the final electron acceptor, combining with electrons and hydrogen ions to form water. This process can generate approximately 32 ATP molecules from one glucose molecule, making it far more efficient than glycolysis alone. In the absence of sufficient oxygen, cells can perform anaerobic respiration or fermentation. Muscle cells, during intense exercise when oxygen delivery cannot meet demand, convert pyruvate to lactate, regenerating NAD+ so glycolysis can continue producing small amounts of ATP. This lactate accumulation contributes to muscle fatigue and the burning sensation during intense exercise. The liver later converts lactate back to pyruvate or glucose when oxygen is available. Yeast and some bacteria perform alcoholic fermentation, converting pyruvate to ethanol and carbon dioxide, a process humans have exploited for millennia in brewing and baking. The synthesis of proteins, essential for virtually all cellular functions, is a complex process involving DNA transcription and RNA translation. Your DNA contains the genetic instructions for building proteins, but these instructions must first be transcribed into messenger RNA. In the nucleus, RNA polymerase binds to a promoter region on DNA and unwinds the double helix. It then synthesizes a complementary strand of messenger RNA using one DNA strand as a template, matching adenine with uracil instead of thymine and guanine with cytosine. This primary transcript undergoes processing, including the removal of non-coding sequences called introns and the splicing together of coding sequences called exons. A 5' cap and poly-A tail are added to protect the messenger RNA and aid in translation. The mature messenger RNA exits the nucleus through nuclear pores and enters the cytoplasm. Ribosomes, composed of ribosomal RNA and proteins, bind to the messenger RNA and begin translation. Transfer RNA molecules, each carrying a specific amino acid and possessing an anticodon complementary to a messenger RNA codon, deliver amino acids to the ribosome. The ribosome reads the messenger RNA codons, and matching transfer RNA molecules bring the corresponding amino acids. The ribosome catalyzes peptide bond formation between adjacent amino acids, gradually building a polypeptide chain. When a stop codon is reached, translation terminates and the completed polypeptide is released. This polypeptide then folds into its functional three-dimensional structure, sometimes with the assistance of chaperone proteins. and may undergo post-translational modifications such as phosphorylation or glycosylation that affect its activity, location, or stability. Gene expression, the process by which genetic information directs protein synthesis, is tightly regulated, allowing cells to respond to internal and external signals. In prokaryotes, operons allow coordinated regulation of multiple genes, as exemplified by the lac operon in bacteria, which controls genes for lactose metabolism. In eukaryotes, gene expression is regulated at multiple levels. Chromatin structure affects gene accessibility, with tightly packed heterochromatin being transcriptionally inactive and loosely packed euchromatin being accessible. Chemical modifications of DNA and histones, including methylation and acetylation, influence gene expression without changing the DNA sequence, a field called epigenetics. Transcription factors, proteins that bind to specific DNA sequences, can activate or repress transcription. These factors integrate various signals to determine when and how much of a gene is expressed. Includes alternative splicing, where different combinations of exons are joined to produce different proteins from a single gene, and regulation of messenger RNA stability and translation. MicroRNAs, small non-coding RNAs, can bind to messenger RNAs and prevent their translation or promote their degradation, providing another layer of control. Cell division is essential for growth, development, and tissue repair. The cell cycle consists of interphase, when the cell grows and replicates its DNA, and mitosis, when the cell divides. Interphase includes G1 phase, during which the cell grows and performs normal functions, S phase, when DNA replication occurs, producing two identical copies of each chromosome, and G2 phase, when the cell continues growing and prepares for division. During DNA replication, the enzyme helicase unwinds the double helix, and DNA polymerase synthesizes new complementary strands, using each original strand as a template. Because DNA polymerase can only add nucleotides to an existing strand, short RNA primers are laid down by primase, and DNA polymerase extends from these primers. On the leading strand, synthesis is continuous, but on the lagging strand, it occurs in short segments called Okazaki fragments, which are later joined by DNA ligase. Multiple proofreading mechanisms ensure high fidelity, with errors occurring in only about 1 in 1 billion nucleotides. Mitosis itself consists of several phases. During prophase, chromatin condenses into visible chromosomes, each consisting of two sister chromatids joined at the centromere. The nuclear envelope breaks down, and the mitotic spindle, composed of microtubules extending from centrosomes, begins to form. In metaphase, chromosomes align at the cell's equator, attached to spindle fibers at their kinetochores. Anaphase begins when sister chromatids separate and move to opposite poles of the cell, pulled by shortening spindle fibers. During telophase, nuclear envelopes reform around each set of chromosomes, which begin to decondense, and the spindle disassembles. Cytokinesis, the division of the cytoplasm, occurs concurrently with telophase. In animal cells, a contractile ring of actin and myosin pinches the cell in two, forming a cleavage furrow. The result is two genetically identical daughter cells. Checkpoints throughout the cell cycle ensure that division proceeds only when conditions are appropriate and previous steps have completed correctly. The G1 checkpoint verifies adequate cell size and nutrients. The G2 checkpoint ensures complete and accurate DNA replication. And the metaphase checkpoint confirms that all chromosomes are properly attached to spindle fibers before anaphase proceeds. Meiosis, a specialized type of cell division occurring only in germ cells, produces gametes with half the chromosome number of the parent cell. This reduction is essential because fertilization combines genetic material from two parents, and without it, chromosome number would double with each generation. Meiosis consists of two consecutive divisions. During meiosis 1, homologous chromosomes pair up in a process called synapsis, forming tetrads. Crossing over occurs, where homologous chromosomes exchange segments of DNA, creating new combinations of alleles and increasing genetic diversity. The homologous pairs then separate, with one chromosome from each pair going to each daughter cell. Meiosis 2 resembles mitosis, with sister chromatids separating, resulting in 4 haploid cells, each genetically unique. In males, meiosis produces 4 functional sperm, while in females, it produces 1 ovum and 3 polar bodies that typically degenerate, ensuring the ovum retains most of the cytoplasm and organelles. Stem cells possess the remarkable ability to divide and differentiate into various cell types, making them crucial for development, growth, and tissue repair. Embryonic stem cells, derived from the inner cell mass of blastocysts, are pluripotent, capable of becoming any cell type in the body. Adult stem cells, found in various tissues, are multipotent, able to differentiate into a limited range of cell types. Hematopoietic stem cells in bone marrow give rise to all blood cell types, while mesenchymal stem cells can become bone, cartilage, muscle, or fat cells. Neural stem cells in certain brain regions can generate new neurons and glial cells throughout life, although neurogenesis in adults is limited compared to development. The signals that direct stem cell differentiation involve growth factors, cell-cell interactions, and changes in gene expression. Understanding these mechanisms has tremendous therapeutic potential for treating injuries and diseases involving cell loss or dysfunction. Apoptosis, or programmed cell death, is a normal and essential process distinct from necrosis, which results from acute injury. Apoptosis eliminates damaged, infected, or unnecessary cells in a controlled manner that does not trigger inflammation. During development, apoptosis sculpts tissues, removing webbing between developing fingers and toes and eliminating excess neurons. In adults, it maintains tissue homeostasis by balancing cell division, removes potentially dangerous cells with DNA damage that could become cancerous, and eliminates immune cells that react against the body's own tissues. The apoptotic process can be triggered by external signals binding to death receptors on the cell surface, or by internal stress, such as DNA damage or lack of survival signals. Either pathway activates caspases, proteolytic enzymes that systematically dismantle the cell, cleaving structural proteins and activating enzymes that fragment DNA. The cell shrinks, its membrane forms blebs, and the nucleus condenses and fragments. Phosphatidylserine, normally on the inner leaflet of the plasma membrane, flips to the outer surface, marking the cell for phagocytosis. Macrophages recognize and engulf apoptotic cells before their contents can leak out and damage surrounding tissue or trigger inflammation. Defects in apoptosis contribute to various diseases, with insufficient apoptosis allowing cancer cells to survive when they should die, and excessive apoptosis contributing to neurodegenerative diseases. Cancer represents a breakdown of normal cellular regulation, arising when cells accumulate mutations that disrupt growth control, apoptosis, and other regulatory mechanisms. Cancer cells exhibit several hallmarks including self-sufficiency in growth signals, insensitivity to anti-growth signals, evasion of apoptosis, limitless replicative potential, sustained angiogenesis, and ability to invade tissues and metastasize. Normal cells require external growth signals to divide, but cancer cells often produce their own growth factors or have mutations in growth factor receptors that keep them perpetually activated. tumor suppressor genes like p53 normally halt cell division when DNA damage is detected, allowing time for repair or triggering apoptosis if damage is irreparable. Mutations inactivating tumor suppressor genes remove these brakes on division. Oncogenes, mutated versions of normal genes called proto-oncogenes, can drive excessive cell proliferation. The ROS protein, frequently mutated in cancers, is a molecular switch in growth signaling pathways, and mutations can lock it in the active state. Telomeres, protective caps on chromosome ends, normally shorten with each cell division, eventually triggering senescence or apoptosis, limiting the number of times a cell can divide. Cancer cells often reactivate telomerase, an enzyme that rebuilds telomeres, allowing unlimited divisions. As tumors grow, they require increased blood supply, so cancer cells secrete vascular endothelial growth factor, stimulating formation of new blood vessels through angiogenesis. These vessels are often abnormal, with irregular structure and leaky walls. Metastasis, the spread of cancer to distant sites, accounts for most cancer deaths. Cancer cells acquire the ability to invade surrounding tissue by secreting enzymes that degrade extracellular matrix, and by losing adhesion molecules that normally keep cells together. They enter blood or lymphatic vessels, survive in circulation, and establish new tumors at distant sites. The immune system can recognize and destroy cancer cells, but tumors evolve mechanisms to evade immunity, such as downregulating antigens that mark them as abnormal or secreting factors that suppress immune responses. Cancer development is a multi-step process requiring accumulation of multiple mutations, which explains why cancer incidence increases with age, as mutations accumulate over time. Environmental factors including tobacco smoke, ultraviolet radiation, certain chemicals, and some viruses increase mutation rates and cancer risk. Genetics also plays a role, with inherited mutations in genes like BRCA1 and BRCA2 substantially increasing breast and ovarian cancer risk. The complexity of metabolism extends far beyond cellular respiration, encompassing all chemical reactions in the body. Anabolic pathways consume energy to build complex molecules from simpler ones, such as protein synthesis from amino acids or glycogen synthesis from glucose. Catabolic pathways break down complex molecules to release energy, as in the digestion of proteins to amino acids or the breakdown of glycogen to glucose. These pathways are regulated to meet the body's changing energy demands. Hormones play crucial roles in metabolic regulation. Insulin, released when blood glucose is elevated, promotes glucose uptake by cells and its storage as glycogen in liver and muscles, and stimulates protein synthesis and fat storage, creating an anabolic state. Glucagon, released when blood glucose is low, stimulates glycogen breakdown and glucose release from the liver, activating catabolic pathways to mobilize stored energy. Epinephrine, released during stress or exercise, rapidly mobilizes energy by stimulating glycogen breakdown and fat release from adipose tissue. Thyroid hormones regulate metabolic rate, increasing oxygen consumption and heat production. Cortisol, released during prolonged stress, promotes gluconeogenesis, the synthesis of glucose from non-carbohydrate sources like amino acids and glycerol, and increases protein breakdown, providing substrates for glucose production. Growth hormone stimulates protein synthesis and growth, while also promoting fat breakdown and opposing insulin's effects on glucose uptake. Leptin, produced by adipose tissue in proportion to fat stores, signals the brain about energy reserves, suppressing appetite when stores are adequate. Ghrelin, produced by the stomach, stimulates appetite, especially before meals. These hormonal signals integrate with neural inputs to regulate food intake and energy expenditure. Lipid metabolism involves synthesis and breakdown of fats. Triglycerides, the main storage form of fat, consist of three fatty acids attached to a glycerol backbone. During fat synthesis, or lipogenesis, excess glucose can be converted to fatty acids in the liver and adipose tissue through a series of reactions requiring ATP and NADPH. These fatty acids combine with glycerol to form triglycerides stored in adipocytes. During lipolysis, triggered by hormones like epinephrine and glucagon, triglycerides are broken down to glycerol and fatty acids. Glycerol can be converted to glucose in the liver, while fatty acids undergo beta oxidation in mitochondria, progressively cleaving two carbon units that enter the citric acid cycle, generating substantial ATP. Ketone bodies, produced in the liver from fatty acids during prolonged fasting or carbohydrate restriction, can be used by the brain and other tissues as alternative fuel. Cholesterol, though often villainized, is essential for cell membrane structure, synthesis of steroid hormones and bile acids, and vitamin D production. The liver synthesizes cholesterol, and dietary cholesterol absorption is regulated. Lipoproteins transport cholesterol and triglycerides in blood. Low-density lipoproteins deliver cholesterol to tissues, but can deposit it in arterial walls, contributing to atherosclerosis. High-density lipoproteins remove excess cholesterol from tissues and transport it to the liver for excretion, providing cardiovascular protection. Amino acid metabolism involves synthesis of non-essential amino acids from metabolic intermediates and nitrogen sources, while essential amino acids must be obtained from diet. Transamination reactions transfer amino groups between molecules, interconverting amino acids and linking amino acid metabolism to carbohydrate metabolism. Deamination removes amino groups from amino acids, producing ammonia, a toxic substance that the liver converts to urea through the urea cycle. Urea, much less toxic than ammonia, is transported in blood to the kidneys for excretion. Some amino acids serve as precursors for important molecules beyond proteins. Tyrosine is used to synthesize dopamine, norepinephrine, epinephrine, and thyroid hormones. Tryptophan is the precursor for serotonin and melatonin. Glutamate serves as a neurotransmitter and as a precursor for GABA. Nucleotide metabolism involves synthesis and degradation of DNA and RNA building blocks. Purines and pyrimidines can be synthesized de novo from amino acids, ribose phosphate, and carbon dioxide through complex pathways requiring substantial energy, or salvaged from degraded nucleic acids, a more economical route. Purine degradation produces uric acid, which is less soluble than urea and can crystallize in joints, causing gout when levels are elevated. The integration of these metabolic pathways allows the body to maintain energy homeostasis across varying conditions. During the fed state, absorbed nutrients are stored, with excess glucose converted to glycogen and fat, and amino acids used for protein synthesis. During the fasting state, stored nutrients are mobilized, with glycogen broken down to maintain blood glucose, and later, as glycogen depletes, fat and protein are increasingly used, with the liver producing glucose from glycerol and amino acids. During exercise, energy demands increase dramatically, and metabolic pathways shift to meet these demands, utilizing muscle glycogen, blood glucose, and fatty acids depending on exercise intensity and duration. The molecular basis of inheritance explains how traits pass from parents to offspring and how variation arises. Mendel's principles of inheritance describe how discrete units of heredity, genes, are transmitted, Each gene has different versions called alleles. In diploid organisms like humans, each cell contains two alleles for each gene, one inherited from each parent. If both alleles are identical, the individual is homozygous for that gene. If different, heterozygous. Dominant alleles mask the effect of recessive alleles in heterozygotes. During meiosis, allele pairs separate, with each gamete receiving one allele, and fertilization restores the diploid state. Independent assortment means alleles of different genes are distributed to gametes independently, although this is modified by linkage when genes are close together on the same chromosome. Crossing over during meiosis can separate linked genes, with recombination frequency reflecting the distance between genes on a chromosome. Many traits show more complex inheritance patterns than simple Mendelian genetics. Incomplete dominance occurs when heterozygotes show an intermediate phenotype, as in snapdragons where red and white alleles produce pink flowers in heterozygotes. Codominance occurs when both alleles are fully expressed, as in the Abo blood group system, where individuals with A and B alleles produce both A and B antigens. Multiple alleles exist for some genes, with more than two versions in the population, though each individual still has only two. Polygenic traits like height and skin color are influenced by multiple genes, each contributing a small effect, producing continuous variation in the population. Environmental factors also influence phenotype, interacting with genotype. Genetic disorders result from mutations, changes in DNA sequence. Point mutations involve a single nucleotide change, which may be silent, producing no change in the protein due to the degeneracy of the genetic code, or may be missense, changing one amino acid, or nonsense, creating a premature stop codon. Sickle cell disease results from a single nucleotide substitution in the beta-globin gene, changing one amino acid in hemoglobin, which causes red blood cells to deform under low oxygen conditions. Carriers, heterozygous for the sickle cell allele, have some resistance to malaria, demonstrating how harmful alleles can persist in populations when heterozygotes have advantages. Deletions and insertions of nucleotides can cause frameshift mutations, altering all downstream codons and usually producing non-functional proteins. Chromosomal abnormalities involve larger-scale changes. Non-disjunction, failure of chromosomes to separate properly during meiosis, produces gametes with abnormal chromosome numbers. Down syndrome results from trisomy 21, 3 copies of chromosome 21. Sex chromosome aneuploidies, such as Turner syndrome with a single X chromosome or Klinefelter syndrome with XXY, demonstrate that the number of sex chromosomes can vary while still permitting survival. Some genetic disorders show sex-linked inheritance because genes are located on sex chromosomes. X-linked recessive disorders like hemophilia and Duchenne muscular dystrophy predominantly affect males who have only one X chromosome, while females with 2 X chromosomes are usually carriers. X inactivation in females, where one X chromosome in each cell is randomly inactivated early in development, equalizes X-linked gene expression between sexes and creates mosaic females who express different X chromosomes in different cells. Genetic testing can identify carriers of recessive alleles, predict disease risk, and diagnose genetic disorders prenatally or after birth. Advances in genomics have enabled genome-wide association studies, identifying common genetic variants associated with complex diseases. The human genome contains approximately 3 billion base pairs encoding about 20,000 genes, though only about 1% of the genome codes for proteins. Much of the non-coding DNA includes regulatory sequences controlling gene expression, sequences with structural roles, and repetitive sequences whose functions are still being discovered. Understanding the molecular biology of the human body reveals the fundamental unity of life, with the same genetic code used by all organisms, the same basic metabolic pathways, and the same cellular structures. This shared heritage reflects common ancestry, with all life on Earth descended from a common ancestor billions of years ago. Evolution through natural selection has shaped the human body, with traits that enhanced survival and reproduction becoming more common over generations. The opposable thumb, bipedal locomotion, large brain, and capacity for language represent key evolutionary adaptations. Vestigial structures like the appendix, tailbone, and wisdom teeth reflect evolutionary history, having reduced in size and function as lifestyles changed. Evolutionary medicine examines how evolutionary history influences health and disease, explaining, for instance, why we crave high-calorie foods. Adaptive when food was scarce, but problematic in modern environments with abundant food. The human body represents the result of millions of years of evolutionary refinement, trillions of coordinated cellular activities, and intricate molecular mechanisms working continuously to maintain life. From DNA replication, ensuring faithful transmission of genetic information, to the immune system defending against countless pathogens, to the brain processing sensory information and generating consciousness, every aspect of human biology demonstrates remarkable complexity and elegant design shaped by natural selection. As you rest, your body continues its ceaseless work, cells dividing and dying, proteins being synthesized and degraded, nutrients being metabolized, waste being eliminated, all coordinated through hormonal and neural signaling, maintaining the stable internal environment necessary for life. Your heart beats, your lungs breathe, your kidneys filter, your liver detoxifies, your immune system patrols, all without conscious effort. A testament to the sophisticated biological systems that sustain your existence every moment of every day. Let us delve even deeper into the magnificent intricacies of human physiology, exploring additional dimensions of how your body functions at the molecular, cellular, and systemic levels. The process of wound healing demonstrates the remarkable coordination between multiple biological systems. When tissue is damaged, whether through a cut, scrape, or surgical incision, the body immediately initiates a complex repair process involving 4 overlapping phases. The hemostasis phase begins within seconds of injury as blood vessels constrict to reduce blood loss, and platelets adhere to exposed collagen fibers, forming a temporary plug. The coagulation cascade activates, with clotting factors interacting in a sequential manner, ultimately converting fibrinogen to fibrin threads that weave through the platelet plug, creating a stable blood clot. This clot not only stops bleeding, but also serves as a scaffold for arriving cells. The inflammatory phase follows, characterized by the classic signs of inflammation including redness, heat, swelling, and pain. Damaged cells release chemical signals including histamine and prostaglandins that cause vasodilation and increase capillary permeability, allowing fluid and immune cells to enter the damaged tissue. Neutrophils arrive first, typically within hours, engulfing bacteria and cellular debris. Macrophages follow, continuing to clean the wound while secreting growth factors that stimulate the next phase. The proliferative phase involves the formation of granulation tissue, a temporary structure rich in new blood vessels, fibroblasts, and collagen. Fibroblasts migrate into the wound and begin synthesizing collagen, primarily type 3 collagen initially, which provides structural support. Angiogenesis, the formation of new blood vessels, occurs as endothelial cells proliferate and organize into capillary networks, delivering oxygen and nutrients essential for healing. Epithelial cells at the wound edges begin to migrate across the wound surface, a process called reepithelialization, gradually covering the exposed tissue. In skin wounds, myofibroblasts, specialized cells with contractile properties, pull wound edges together, reducing the wound size. The remodeling phase can last months to years, as the temporary granulation tissue is gradually replaced with stronger, more organized tissue. Type 3 collagen is replaced with type 1 collagen, which is arranged along tension lines, increasing tissue strength. Excess cells undergo apoptosis, and the tissue gradually regains function, though healed tissue rarely achieves the full strength of undamaged tissue, typically reaching about 80% of original strength. Scar tissue formation represents the body's compromise between rapid healing and perfect restoration. Factors influencing wound healing include adequate nutrition, particularly protein, vitamin C, and zinc, sufficient blood supply delivering oxygen and nutrients, absence of infection which can prolong inflammation and damage tissue, and various systemic factors including age, diabetes, and certain medications. Chronic wounds, such as diabetic ulcers or pressure sores, result from impaired healing processes and pose significant medical challenges. The aging process affects all body systems, representing accumulated changes at molecular, cellular, and organ levels. At the cellular level, telomere shortening with each division eventually limits replicative capacity, contributing to tissue aging. Senescent cells, which have stopped dividing but remain metabolically active, accumulate with age and secrete inflammatory molecules that can damage surrounding tissue. Oxidative damage from reactive oxygen species, byproducts of normal metabolism, accumulates over time, damaging proteins, lipids, and DNA. Mitochondrial function declines with age, reducing cellular energy production. DNA damage accumulates as repair mechanisms become less efficient and mutations increase. Protein homeostasis deteriorates as synthesis slows and degradation of damaged proteins becomes less efficient, leading to accumulation of misfolded proteins that can aggregate and disrupt cellular function. Epigenetic changes alter gene expression patterns without changing DNA sequence, affecting cellular function. At the tissue and organ level, aging manifests in numerous ways. Skin loses elasticity as collagen and elastin degrade faster than they are replaced, and subcutaneous fat diminishes, contributing to wrinkles and sagging. Bone density decreases, particularly after menopause in women due to declining estrogen levels, increasing fracture risk. Osteoporosis, characterized by porous, fragile bones, affects millions of older adults. Sarcopenia, the age-related loss of muscle mass and strength, results from decreased protein synthesis, reduced physical activity, hormonal changes, and motor neuron loss. Joints experience wear on articular cartilage, which has limited regenerative capacity, potentially leading to osteoarthritis. The cardiovascular system shows increased arterial stiffness as elastic fibers in vessel walls degrade and are replaced with collagen, raising blood pressure. The heart muscle may thicken and maximum heart rate declines. Atherosclerosis, the accumulation of plaques in arterial walls, progresses with age, though lifestyle factors significantly influence its rate. The respiratory system experiences reduced lung elasticity and chest wall compliance, decreasing vital capacity. Alveolar surface area diminishes slightly, and respiratory muscle strength declines, making older adults more vulnerable to respiratory infections. The nervous system undergoes subtle changes, with some neuron loss in specific brain regions, though most cognitive functions remain intact in healthy aging. Processing speed may slow, and forming new memories may take longer, though accumulated knowledge and wisdom often compensate. The risk of neurodegenerative diseases, including Alzheimer's and Parkinson's, increases with age. Sensory systems decline with presbyopia, the inability to focus on near objects, affecting nearly everyone by middle age as the lens loses flexibility. Hearing often declines, particularly for high frequencies, due to hair cell loss in the cochlea. Taste and smell sensitivity may decrease, potentially affecting appetite and nutrition. The immune system shows immunosenescence, with decreased production of new lymphocytes, reduced antibody responses to new antigens, and increased autoimmune tendencies. This makes older adults more susceptible to infections and less responsive to vaccinations. Endocrine changes include declining growth hormone, sex hormone, and DHEA levels. In women, menopause marks the end of reproductive capacity as ovarian follicles are depleted and estrogen production drops dramatically. In men, testosterone levels gradually decline, though sperm production continues into advanced age. Despite these changes, aging is highly variable among individuals, influenced by genetics, lifestyle factors including diet, exercise, stress management, social engagement, and environmental exposures. The concept of successful aging emphasizes maintaining function and quality of life, rather than merely extending lifespan. Exercise physiology reveals how physical activity profoundly affects the body. During exercise, energy demands increase dramatically, requiring coordinated responses from multiple systems. The cardiovascular system responds with increased heart rate and stroke volume, raising cardiac output to deliver more oxygen-rich blood to working muscles. Blood flow is redistributed, with vasoconstriction in non-essential organs and vasodilation in muscles, increasing their blood supply from about 20% at rest to up to 80% during intense exercise. The respiratory system increases breathing rate and depth, enhancing oxygen intake and carbon dioxide removal. At the cellular level, muscle contractions are powered by ATP, which is regenerated through 3 energy systems used in sequence, depending on exercise intensity and duration. The phosphagen system provides immediate energy by transferring a phosphate group from creatine phosphate to ADP, regenerating ATP very rapidly, but only for about 10 seconds of maximum effort. The glycolytic system breaks down glucose or glycogen without oxygen, producing ATP quickly, but also generating lactate. This system dominates during high-intensity exercise, lasting from about 10 seconds to 2 minutes. The oxidative system uses cellular respiration to produce ATP from carbohydrates, fats, and sometimes proteins. This system produces ATP more slowly, but can sustain activity for extended periods, dominating during low to moderate intensity exercise. During moderate exercise, fat oxidation provides much of the energy, with carbohydrates becoming increasingly important as intensity increases. At very high intensities, carbohydrate becomes the predominant fuel because it produces more ATP per oxygen molecule than fat, though carbohydrate stores are limited. Regular exercise training produces numerous adaptations. Cardiovascular adaptations include increased stroke volume, allowing the heart to pump more blood with each beat, decreased resting heart rate as the heart becomes more efficient, increased capillary density in muscles improving oxygen delivery, and potential increases in maximal cardiac output. These changes improve cardiovascular efficiency and endurance. Muscular adaptations depend on training type. Endurance training increases mitochondrial density and oxidative enzymes in muscle fibers, enhancing aerobic capacity. It promotes conversion of fast glycolytic fibers toward more oxidative types. Resistance training stimulates muscle protein synthesis, increasing muscle fiber size through hypertrophy. Motor unit recruitment becomes more efficient, and tendons and ligaments strengthen. Bone responds to mechanical loading by increasing density, reducing osteoporosis risk. Metabolic adaptations include improved insulin sensitivity, better glucose control, increased fat oxidation capacity, and favorable changes in blood lipid profiles, with increased HDL and decreased triglycerides. The nervous system shows improved neuromuscular coordination, enhanced motor unit recruitment, and increased efficiency of movement patterns. Exercise also benefits mental health through multiple mechanisms. Physical activity stimulates release of endorphins, naturally occurring opioids that produce feelings of well-being. It increases brain-derived neurotrophic factor, which supports neuron survival and promotes neurogenesis in the hippocampus, potentially enhancing memory and learning. Exercise reduces stress hormones like cortisol, while increasing production of neurotransmitters, including serotonin and norepinephrine that regulate mood. Regular physical activity is associated with reduced risk of depression and anxiety, improved sleep quality, and enhanced cognitive function, including in older adults where it may reduce dementia risk. The acute exercise response differs from training adaptations. During a single bout of exercise, heart rate and breathing increase, body temperature rises, and metabolic rate elevates. Recovery involves gradual return to baseline, with heart rate and breathing normalizing within minutes to hours depending on intensity. Excess post-exercise oxygen consumption, sometimes called the afterburn effect, means metabolic rate remains elevated for hours after exercise as the body replenishes energy stores, repairs tissue, and restores homeostasis. Sleep represents another fundamental physiological state, essential for health and survival. Sleep architecture consists of cycles through different stages, each with distinct brain wave patterns, physiological characteristics, and functions. Non-REM sleep includes 3 stages. Stage 1 is a brief transition from wakefulness, lasting only a few minutes, characterized by theta waves and easily disrupted. Stage 2, comprising about half of total sleep time, shows theta waves with intermittent sleep spindles and K-complexes, thought to be involved in memory consolidation and sensory gating. Stage 3, slow-wave sleep or deep sleep, features delta waves, high-amplitude, low-frequency brain activity. This stage is most restorative when growth hormone is released, tissue repair occurs, and immune function is enhanced. It is difficult to awaken someone from slow-wave sleep. REM sleep, or rapid eye movement sleep, is characterized by brain activity resembling wakefulness, rapid eye movements, vivid dreaming, and muscle atonia. Paralysis of voluntary muscles except the diaphragm and eye muscles, preventing dream enactment. REM sleep appears crucial for emotional regulation, memory consolidation, particularly for procedural and emotional memories, and possibly creativity. The proportion of REM sleep increases with each cycle through the night. A typical night includes 4 to 6 sleep cycles, each lasting about 90 minutes, with more slow-wave sleep in early cycles and more REM sleep later. Sleep is regulated by 2 processes: homeostatic sleep pressure, which builds during wakefulness as adenosine accumulates in the brain, and the circadian rhythm, the internal biological clock synchronized to the light-dark cycle. The suprachiasmatic nucleus in the hypothalamus serves as the master circadian pacemaker, receiving light information from the retina via the retinohypothalamic tract. It coordinates circadian rhythms throughout the body through neural and hormonal signals, including regulating melatonin release from the pineal gland. Melatonin secretion increases in darkness, promoting sleepiness, and decreases with light exposure. Sleep serves multiple critical functions. It is essential for memory consolidation, with different sleep stages contributing to consolidation of different memory types. Sleep supports immune function, with sleep deprivation impairing immune responses and increasing infection susceptibility. Metabolic regulation depends on adequate sleep, with sleep restriction associated with insulin resistance, increased appetite through altered leptin and ghrelin levels, and weight gain. Brain clearance of metabolic waste products, including beta amyloid associated with Alzheimer's disease, occurs primarily during sleep through the glymphatic system, where cerebrospinal fluid flows through brain tissue, clearing waste. Sleep deprivation has numerous negative effects including impaired cognitive function, reduced attention and reaction time, decreased emotional regulation, increased stress hormones, elevated blood pressure, and over time, increased risk of obesity, diabetes, cardiovascular disease, and neurodegenerative disorders. Chronic partial sleep deprivation, common in modern society, accumulates sleep debt that cannot be fully repaid with occasional extended sleep. Individual sleep needs vary, with most adults requiring 7 to 9 hours nightly, though genetics influence both sleep duration needs and chronotype. Whether someone is naturally a morning or evening person. Sleep disorders are common and include insomnia, difficulty falling or staying asleep, sleep apnea, where breathing repeatedly stops and starts during sleep, often due to airway obstruction. Narcolepsy, characterized by excessive daytime sleepiness and sudden sleep attacks, and restless leg syndrome, causing uncomfortable sensations and an urge to move the legs, disrupting sleep. Circadian rhythm disorders occur when the internal clock is misaligned with external demands, as in shift work or jet lag. The stress response, mediated primarily through the hypothalamic pituitary adrenal axis and the sympathetic nervous system, prepares the body to deal with perceived threats. When the brain perceives a stressor, the hypothalamus releases corticotropin-releasing hormone, which stimulates the anterior pituitary to secrete adrenocorticotropic hormone. This hormone travels through blood to the adrenal cortex, stimulating cortisol release. Cortisol, the primary stress hormone, mobilizes energy by promoting glucose release, increases blood pressure and cardiovascular function, suppresses non-essential functions including digestion and reproduction, and modulates immune function. Simultaneously, the sympathetic nervous system activates, with the adrenal medulla releasing epinephrine and norepinephrine, triggering the fight-or-flight response. Heart rate and blood pressure increase, airways dilate, pupils dilate for better vision, digestion slows as blood is redirected to muscles, and glucose is released for immediate energy. This acute stress response is adaptive for short-term threats, enabling rapid physical responses to danger. However, chronic stress, where the stress response remains activated for extended periods, has detrimental health effects. Prolonged cortisol elevation can suppress immune function, increasing infection susceptibility, impair memory and learning through effects on the hippocampus, contribute to anxiety and depression, promote abdominal fat accumulation and insulin resistance, accelerate atherosclerosis, and disrupt sleep. Chronic stress is associated with increased risk of numerous health problems including cardiovascular disease, autoimmune disorders, gastrointestinal problems, and mental health disorders. Stress management techniques can mitigate these effects. Regular exercise reduces stress hormones and stimulates endorphin release. Mindfulness and meditation practices can reduce activity in stress-related brain regions and lower cortisol levels. Adequate sleep supports stress resilience. Social support provides emotional buffering against stress. Time management and problem-solving skills can reduce perceived stress by increasing sense of control. The gut microbiome, the community of trillions of microorganisms inhabiting the digestive tract, particularly the large intestine, has emerged as a crucial factor in health. This ecosystem includes bacteria, archaea, fungi, viruses, and other microbes, with bacterial species numbering in the thousands. The composition of the microbiome is influenced by genetics, mode of birth delivery, infant feeding, diet, medications, particularly antibiotics, and environmental exposures. The microbiome performs numerous beneficial functions. Microbes ferment indigestible dietary fibers, producing short-chain fatty acids including butyrate, which serves as the primary energy source for colonocytes, the cells lining the colon, and has anti-inflammatory properties. The microbiome synthesizes certain vitamins, including vitamin K and some B vitamins. It trains and regulates the immune system, with microbial molecules stimulating immune development and maintaining the balance between immune activation and tolerance. The microbiome protects against pathogens through competitive exclusion, occupying ecological niches, and producing antimicrobial substances that inhibit pathogen colonization. It influences metabolism, affecting energy harvest from food, fat storage, and insulin sensitivity. Emerging research reveals the gut-brain axis, bidirectional communication between the gut and brain mediated through neural pathways including the vagus nerve, hormonal signaling, and immune-mediated pathways. The microbiome produces neurotransmitters and neurotransmitter precursors, including serotonin, dopamine, and GABA. Though whether gut-produced neurotransmitters reach the brain remains debated. Microbial metabolites can influence brain function through various mechanisms. Studies link altered microbiome composition to conditions including obesity, diabetes, inflammatory bowel disease, allergies, and mental health disorders including depression and anxiety. Though establishing causation remains challenging. Dysbiosis, an imbalance in the microbiome composition, may contribute to disease, though whether dysbiosis causes disease or results from it varies by condition. Maintaining a healthy microbiome involves consuming a diverse, fiber-rich diet with prebiotics, non-digestible compounds that promote beneficial microbe growth, and fermented foods containing live microbes acting as probiotics. Avoiding unnecessary antibiotics helps preserve microbiome diversity. The concept of biological rhythms extends beyond circadian rhythms. Ultradian rhythms, cycles shorter than 24 hours, include the 90-minute sleep cycles, the nasal cycle where nostril dominance alternates, and rhythms in hormone secretion and alertness. Infradian rhythms, longer than 24 hours, include the menstrual cycle, averaging 28 days, during which hormonal fluctuations orchestrate ovarian and uterine changes. The follicular phase begins with menstruation, when the uterine lining is shed, and continues as rising FSH stimulates follicle development and estrogen secretion. The luteal phase follows ovulation, with the corpus luteum secreting progesterone that maintains the endometrium for potential implantation. If pregnancy does not occur, the corpus luteum degenerates, hormone levels fall, and menstruation begins a new cycle. Seasonal rhythms affect various physiological parameters, with some people experiencing seasonal affective disorder, depression associated with reduced daylight in winter months, linked to altered melatonin secretion and circadian disruption. Thermoregulation maintains core body temperature around 37 degrees Celsius despite environmental variations. The hypothalamus acts as the body's thermostat, receiving input from thermoreceptors in skin and internal organs. When body temperature rises, heat loss mechanisms activate, including vasodilation of skin blood vessels, increasing heat radiation from the body's surface, and sweating, where evaporation of sweat removes heat. Behavioral responses include seeking cooler environments and removing clothing. When body temperature falls, heat conservation and generation mechanisms activate. Vasoconstriction of skin blood vessels reduces heat loss. Shivering, rapid involuntary muscle contractions, generates heat through increased metabolic activity. Non-shivering thermogenesis occurs in brown adipose tissue, specialized fat tissue rich in mitochondria that can generate heat without shivering, particularly important in infants. Behavioral responses include seeking warmth and adding clothing. Fever, an elevated body temperature set point, occurs when pyrogens, substances released during infection or inflammation, act on the hypothalamus to raise the temperature set point. The body then employs heat-generating mechanisms to reach this higher temperature. Fever may enhance immune function and inhibit pathogen growth, representing an adaptive response to infection, though very high fevers can be dangerous. The integration of all these systems, from molecular mechanisms to organ systems, from development through aging, from homeostatic regulation to responses to challenges like exercise, stress, and infection, creates the living, breathing, conscious being that is you. Every heartbeat, every breath, every thought emerges from the coordinated activity of countless cells, the precisely regulated expression of thousands of genes, the balanced interplay of numerous hormones and neurotransmitters, all shaped by billions of years of evolution, and your unique genetic inheritance and life experiences, working together seamlessly to maintain life and enable you to interact with and experience the world around you.